Justin, welcome on the show.
Thank you.
Thank you for giving up the time to speak to me today. Really appreciate that. I know you're very busy and you're flying out tomorrow night, so I really appreciate that. So yeah, last week I came to your event about circadian rhythm, and yeah, I was really surprised about all the background players that take effect on how we feel, all these effects. So tell us a little bit about it, about your your research and also other people's research in this sort of topic.
So it all, my version of the story, it all begins with fruit flies. Yeah. And that's how I got into it, because I was thinking about what to research, and I'd always thought that sleep was really interesting. And I was also really keen to try and get into the kind of genes underlying behavior. So that was where it merged with me, and I discovered this field of circadian rhythms existed as a research area, which I didn't know beforehand. And what was fascinating is people were just pulling out the first few genes and beginning to understand how they work together to drive behavior. Okay. If you go back, the idea that a single gene could have a really big effect on behavior has quite a profound implications. We like to think about free will and everything, but then we've got genes kind of pulling us around in all different ways. And you could even, at its broadest, you could imagine, hopefully not, but a criminal defense, and I didn't do it, my genes made me do it.
Yeah, that's interesting.
At its broadest.
Yeah.
But so this circadian rhythms, these daily rhythms seem like a really interesting way for me to get into that field. And I've actually kind of stuck with that field as well as learning and teaching lots of other behaviors. But this is such a rich area of research that I've stayed with.
How much do we know about this topic? Because it is governing a lot about, a lot of our lives. How much do we really understand the intricacies and the, all the little details of this topic?
So we have a very good understanding of what happens in a single cell. We know, I'd say, 15 to 20 genes that function together to make what we call a molecular clock. So they're keeping time, they're keeping 24-hour time. What we're still trying to understand is exactly what those clocks do in those cells. So the clock in general, if I look at the molecular clock, the circadian clock in a liver cell or even in a fruit fly brain cell, they're quite similar, but obviously they're going to regulate different processes. And so that's what some of the more exciting research now is going on, is understanding what are these clocks doing in different cells of our body, and then how they talk together to give us this internal overall sense of time.
So in terms of, let's say, percentage of, not understanding and understanding it fully to apply it to human beings and how we live, and ultimately, somehow optimize the way we live, where are we in that
I would
scale?
say we're on the more understanding it side.
Yeah.
Because what we've realized is that essentially what the clocks are doing is regulating the function of that cell. We don't always understand why. Yeah. But then, if your liver is important in metabolism and detoxification, turns out that that's under a 24-hour rhythm. And so all the functions of the liver are regulated, or not all, but many of the functions of the liver will be regulated with a rhythm. Applying that is the more challenging thing, because you need to know, perhaps if you want to interfere with some pathway, with a drug, what exactly that pathway is it under a rhythm, when is the peak time to try and interfere with it, and then overall on the body, in terms of overall metabolism, the body gets much more complicated, especially in humans, where it's harder to do the kinds of experiments you can do in simpler systems like mice or fruit flies.
And I remember from your talk you said that the reason why you use flies and mice and flies is because they're a lot easier to test on. But how much can you take from flies and apply it to human beings? Because humans are much more, I'd assume, much more sophisticated and complex.
We are.
Yeah.
Yeah, we are. But they can fly and we can't.
Yeah.
So, in general, things are pretty well conserved.
Yeah.
And so one of the best examples is actually this first clock gene called period. So people had no idea how animals kept a sense of 24 hours, how they kept their circadian time. And so the very early experiments at the end of the 60s into the 70s looked for flies that no longer had 24-hour rhythms. And they identified mutants in a gene called period because it had such a big effect on the fly's circadian period length. So different mutants of those either lost the clock completely, they became behaviorally arrhythmic, or they had 29-hour days or 19-hour days. And all of those turned out to be mutations, different mutations in one gene called period. And so you step forward many years, when it was cloned in flies, and then people started looking, were there similar genes in humans? And no one could find them for a long time. But then, when the human genome was being sequenced and people were starting to put it together and figuring out where in the genome there's a gene and where there's just kind of empty space, they found 3 of these genes that looked like the fly period gene. And if we hadn't known about the fruit fly gene, we would have had no idea what the function of these genes was in humans.
Yeah.
And so it's actually kind of directly from those basic studies in flies that we've understood about these clock genes in humans. Of course, there's more of them. There's 3 of them as opposed to 1. But they seem to serve a very similar function.
Yeah. What about in terms of, let's say, intermittent fasting? I've heard the more evolutionary side of this argument of, when we were, cavemen, we could only eat in a certain period of time because this and that. But now from your research, does that argument hold up as to why because from your, presentation, I found that when you eat in a time-restricted manner, you're more optimally used to lose weight, sleep better, and a lot of the processes we do daily. So what's your thoughts on that?
So just quick for the record, that's not my research.
Okay,
that's,
yeah.
It's mainly one of my colleagues, Sachin Panda, who works at the Salk. But I'm a big fan of his work because he's really taken some of the basic studies and then applied it to look at other organisms. It's basic studies in mice and flies and applied it to look in humans. So the big finding that they found was a big difference between mice eating round the clock and mice eating just for 8 hours a day. And eating, those 8 hours were in their waking time. And they could give them crazy diets that make the mice eating any time of day put on massive amounts of weight. Or that same diet, same calories, the same overall food intake, but restricted to 8 hours a day, so that's the intermittent fasting, they don't put on weight. And that kind of actually blows up this whole idea of calories to some extent, right? Because those mice had eaten the same amount, and yet they're totally different weights.
Is there nuance in there, or is that very clear-cut of the difference?
Very clear-cut. Yeah, I mean, you look at... they let the experiment run for 20 weeks, so 5 months, and I forget the percentage change, but if you look at, you've got a regular-looking mouse and you've got this obese mouse. And in that obese mouse, it's got fatty liver disease, it looks diabetic or pre-diabetic. I mean, it's a mess.
Yeah.
So then they tried to say what happens in humans. And first of all, they had to measure when a human's eating, and we typically think, I have breakfast at 8 a.m. and dinner at 7 p.m. and I'm fine. I'm within that 8 to 10 to 12 hours that seems to be safe for mice. But actually humans we tend to stretch out our days with late night snacks or a little drink late at night or ice cream. And so many of us as a human population are eating 15 or more hours a day.
Jeez.
And so then when you start to restrict the time that so you do kind of intermittent fasting or time restricted eating, for those people they often lose a good few kilos quite quickly, and they can keep it off. So
it is, because I want to hear about the nuance and the variability of this type of research. So because I've heard my friends do it as well. They only eat in an 8-hour window. And they say they've lost a lot of weight, but with others, they do it, and I'm not sure how committed they were, but it didn't really work on them And they even got more hungry and tired and lethargic. And I was like saying, okay, like, is this a general statement, or does this work on certain chronotypes?
Yeah, so that's interesting, right? So you could imagine, right? The idea then would be to eat when your liver is most functional, which is the day, but the day is pretty general. And so if some of us are morning larks or night owls, the peak of liver activity could actually shift a little bit. And so I guess in a most nuanced way, you would want to align your chronotype, your internal time, with all your meals and everything, so you're going to metabolize everything most efficiently.
How does one understand their chronotype? Like, well, basically, maybe explain what chronotype is.
So chronotype literally means, in the same way we have blood type or anything like that, we have a chronotype where chrono is time. And the simplest way it's measured is by getting the mid phase of your sleep. So if you sleep typically, and you have to do this for several weeks to figure it out fairly accurately, but if you sleep from midnight to 8 a.m., then your chronotype is going to be roughly 4.
Okay.
The mid phase of sleep. And that's when you're, but there's a distribution, and it changes with age. I mean, young children are going to be much earlier than that, even if they're sleeping for long times. Yeah. Teenagers go a little bit later, but then there's variability even if you look across people of a similar age. So I think we're really just beginning this, trying to understand the best time to eat for an individual. I mean, this is part of what I would call a hopeful shift in medicine, right? We talk about personalized medicine, and this would be one feature of it. What is the best time for an individual to eat, to take their drugs? That chronotype can even apply to your athletic time. So people that wake up early morning, they tend to have it's pretty constant athletic time peak athletic time during the day. But if you're really a late chronotype, that early morning time is really hard time to exercise, and you're well below your maximum. And this is related, the idea is this is related not to your alertness, but actually to your muscle activity, to the clocks in your muscles driving, being able to function at their maximum. But at this point, I would say, we're into a gray area of what actually is happening in the muscle. I certainly don't know. Some people might. But this chronotype does affect a lot of different bodily functions.
So now we can understand, like, what's the best time to eat, what's the best time to work out, and that, like, raises the question of, like, we're designing our life to be, the most optimal, productive performance. But in terms of sleep, though, how would your chronotype affect sleep, or how would your body, your muscles, liver, etc., how does that contribute to sleep? Because I know when, living on a university, it's like you sleep very late, you sometimes wake up early, and then the whole day you just you're very phased, you're very muddled, you can't concentrate as sharp. But then contrary to the days where you've had sleep, you just feel a lot sharper, you don't need coffee. How does that play, and what's the best time, or all that stuff?
That's a lot of questions in one. That's fine. But I think it's interesting because in the same way that there are differences in chronotype, different people seem to need different amounts of sleep. Yeah. Right. So the average everyone says is 8 hours, but there are plenty of people that would do totally fine with a bit less, and there are plenty of people that seem to need a bit more than 8 hours. So again, it's like height. We have these curves, we have these variations and everything. Exactly how that is controlled genetically is not so clear, although there's one gene I know of which is really associated with short sleepers, people that typically do very well on quite short sleep, 4 to 6 hours, but that's fairly rare. In terms of putting everything together, you then have both your kind of sleep need, which is going to change as an adolescent. That's going to be higher in your kind of late teenage years, early 20s. And when you're doing everything, one thing I'm grateful of at NYU Abu Dhabi is classes don't start quite as early as they used to. So everything in the UAE starts really early. A lot of high schools will start at 7:30, 7:45. And when I first started teaching here, classes started at 8:30. And now the first class on a typical semester is 9.
Yeah.
Maybe it was even earlier than 8:30. But it just seemed, especially for these kind of teenagers who are shifted a little late in their chronotype, a little bit inhumane and not ideal. So what students are you going to get in your 8:30 a.m. class? You could take the same people and they'll be high functioning by 10 a.m., but 8:30 it's more of a struggle to get going.
Yeah. But how about you mentioned how schools are designed, and especially in the States and in some parts of New Zealand as well, they start very early, and they end around 3:30, 4. How would you discussed it, the timing of your school, when your school starts, really dramatically affects your grades they achieve. And how can authorities or public policy makers change this, or what would be the best solution to it?
Right. So let's start with the biology, which is essentially that the teenage body needs more sleep, and it seems to shift a little bit later, almost as if the clock is running a little bit slow. So you wake up your ideal time of waking is going to be a couple of hours, a few hours later than when you become an adult. Same person. So asking any teenagers to function effectively at 7 or 8 in the morning is like asking an adult person to function really, really well at 5 or 6 a.m. A few people would do it, but it's not the time we would choose to learn. So there was a study in Seattle where they shifted the school start time just by 1 hour. And they showed that the teenagers were getting more sleep, which we all know is beneficial to mood, health, growth, all of that stuff. But what happened then was that actually many people, the grades went up, and people were late much less, and people were absent much less. The question of what's the ideal time has to come as a balance between obviously the students, who should be the primary concern, but also the teachers, who have to function, right? So you can't have them working way off their ideal alertness. So it looks like a 1 hour to 2 hour shift could be phenomenal in terms of learning and wakeness. On the other hand, you've got to balance that with maybe parents helping getting their kids off to school and the work life. So I wouldn't want my teenagers waking up by themselves. I happily, or I help them get out of the door in the morning just to make sure they get the day off to start right.
But don't you think it's kind of sort of like an unfair advantage if a school starts late and then another school starts early? Because statistically, the grades of the school starting earlier, they wouldn't do as well as school starting later. Whereas when they're applying for scholarships or grants or etc., does this not beg the question of unfairness or unequitable differences?
But schools are already inequitable. I mean, you have different teachers, different head teachers that set the tone for a particular school. These schools are in different socioeconomic parts of town, so there are already all kinds of inequities. I think if you just... So I guess the question is this going to be up to the schools to choose, or is this something that would happen at a district or even universe level?
Because
whole country.
If you think about a lot of the opportunities students miss out on is typically to do with their grades, and they're not meeting a certain standard. Yet, if we talk about the structures designed, how are they designed, and are they optimally designed for a student to do well, or has it just been designed that way and is kept that way because of tradition and standards?
I would say it seems to be the tradition and the standards. And certainly in the US, high schools tend to start earlier than elementary schools. And often the children are traveling from further away, which exacerbates that even more. One of the interesting things, so in this study in Seattle, in a fairly affluent society, fairly affluent high school where the students were of a higher socioeconomic class, that actually didn't change the absences and tardinesses of the kids going to school. But in a poorer school, those kids were coming up to the same level as the kids from the more affluent school or the more affluent neighborhood. So in a way, having a later start time could actually be an equalizer across socioeconomic status and give the kids from a poorer background higher chance of success. But I agree. I mean, it would be a very complicated algorithm to try and look at, would you keep the kids from the more affluent neighborhood at an earlier start time to rig it in the opposite direction? I don't think so. So it has to be a balance between all these factors, and again, the adults in the room, who the teachers who need to be there during the daytime. It's complicated, and it's hard to change these systems that are set in stone. But the Seattle School District was happy to experiment with this year, with this change by 1 hour. And I have to say, I don't know after that 1 year what they did, but the evidence is pretty clear that the later start time was benefiting students. So I hope they kept it that way.
And is there like a rhythm or a time clock on your learning? You know, when is the best and optimal time to learn? And does it cycle through? You know, is there a cycle to which time you learn best and which time you should just stop learning?
So again, there are optimal times for alertness, and there are optimal kind of sleepy times. I mean, the mid-afternoon is an obvious kind of lull. There's a dip in the alertness coming from your circadian clocks. Having said that, there's also going to be variability from person to person. What you eat can kind of accentuate that mid-afternoon slump, or minimize it depending on what you eat. And I don't know the details, but I know I've never figured it out. But there are days when I really crash in the afternoon, and there are days when I'm just fine. So it's complicated, but most people, massive generalization here, but most people, again, it's different between adults and teenagers, but I would say adults would generally go for that morning time. Teenagers probably shift a little bit later because their morning, they're not quite as alert yet. They haven't fully come out of sleep till a bit later.
And in terms of like productivity, especially when employers are looking at how the employees are being productive and at which times they're productive and which times they're slacking. Because I heard a recent change in, I think, Finland or some of these Norwegian countries, they shortened their workday.
Right.
Because they thought that employees would be that productive regardless of increasing it or decreasing it, keeping it that. So how does that affect it, or would it affect our productivity?
Yeah, so I don't know if that's a circadian clock thing or just a motivation thing. If you know that you're only, I guess, right, at some point as you're doing a task, you do become less effective. I mean, this is the classic examples of doctors. When they're training, they go into these 24-hour shifts.
Yeah.
They do pretty well for 12 hours, and then there's a real kind of split. And some of them keep doing well and making good decisions, but some of them really don't do well after 12 hours. So again, there are individual differences that I wouldn't say we really understand yet. And I don't even know if the same person can function for 24 hours regularly, or if it's more of a random thing, whether they're making good decisions in that second part of the day. But these long shifts are probably pretty unproductive.
Yeah.
The other piece of it is shift work itself, where people are then actually working at nighttime, for example, completely out of line with their light day cycles. And that's really hard because...
Is that not harmful?
So the World Health Authority recently categorized shift work as a likely carcinogen. Jeez. And that has to do with the way that your tissues are metabolizing the things that you take into your body. Again, I don't think we fully understand why that is, but that's the evidence. It's high incidences of certain tumors that I've forgotten now, but that in shift workers, airlines are a separate issue, but the shift workers in particular. But again, it's complicated because often shift workers are from a lower socioeconomic group, maybe not eating so well, so it's a complicated analysis. But the World Health Authority was clear enough that they categorized this as potential carcinogen, which, I mean, that's what we think with, sunlight, some of the food, smoking, exactly. And now we've got shift work, and that's definitely not in most people's thinking.
How about in terms of, you mentioned when you're performing an operation, the likelihood of, I read it somewhere in a book a couple of years ago, but now when you repeat it again, I was like, wow, when you have your operation, there's a likelihood, a change of probability of the success rate.
Yeah. So this was a study on heart surgery, so a very invasive surgery with already high risks. And what was nice about the study is they took the same group of doctors and looked at the success rates of people in the morning and the afternoon. And you would typically think, from what I was just saying about alertness, that you would want to go and have heart surgery in the morning when the doctor's at their most alert and less likely to make a mistake. But it turns out that if you had your heart surgery in the afternoon, on average you had a better prognosis of surviving and less major issues with your heart. And that comes down to the clock in the heart, which is a muscle, is better at repairing itself in the afternoon. So presumably engages those processes more quickly after the surgery than if you had surgery in the morning. Again, we don't really understand it in depth, but it's counterintuitive, so it doesn't seem like it's the doctor's alertness. They're not sleepy in the afternoon making mistakes. They're actually, or maybe they are, but the patients are still doing better. And it's not like everything in the afternoon. In the morning, there's one study saying certain flu vaccines are much more effective in older people, where you want to boost their responses to flu because they're more vulnerable, if you have the vaccination in the morning. And that presumably has to do with the clock in those immune cells being better able to make a functional response when they get the vaccination in the morning. So it actually makes it complicated for a hospital. Yeah, damn.
And
if you're a heart surgeon, you only can work for 3 hours a day.
Yeah,
that's not ideal. But it's all these things, these are actually quite big effects and quite easy to change. This goes back to the diet. We're not saying you have to eat only lettuce or only carrots or whatever for the rest of your life. People are allowed to eat pretty much whatever they want, but just in a restricted time window. And that seems like a more sustainable change for people to make.
But 8 hours, that's a...
The eating? So more of the...
It's very hard.
So the original mice study was 8 hours, and it was 8 hours because that was simple for the experimenter to set up. They could go in the morning, give the mice the food, wait 8 hours, take the food away, go home. So that was why it was 8 hours. So then they wanted to know, is there something magic about 8 hours? Or is 9 hours, 12 hours, 15 hours? Where's the cutoff? And the biggest difference was between 12 and 15 hours. That 12 looked to be on the kind of safe side, whereas by 15 hours of eating, then the mice were gaining a lot of weight. It wasn't as much as with 24 hours, but the biggest difference was between 12 and 15. So when they started the human time-restricted eating, they let people choose a 10 to 12 hour window, which is a little bit more humane. And I don't think any of this is black and white. And some of the studies have looked at what happens if you do 5 days of time restricted eating and 2 days of whenever you want, kind of mimicking maybe the routine of the week, work week, and then the weekend you can do pretty much whatever you want. And many of the benefits are retained with that 5 days of time restricted eating.
So it's a lot more complicated than it's often presented, and like often we perceive it to be as well.
Well, we also want kind of a one rule for everyone.
That's true.
So that's where we're forgetting about the enormous variation we have, both within our own lives as we kind of grow and age, things change, and across the population. I mean, we're fine with accepting that variation in height, but we don't think about it in other medicine, for example.
In terms of, you mentioned personalized medicine. How prevalent is that going to be in the next 10, 15, 20 years? Are we going to see more of that, or what is it going to be like?
I think so. I think I mean, one of the biggest thing will be genome sequences. And that's a whole nother podcast. But that's going to happen more and more. More and more people already have an idea of their genetic susceptibilities, the likelihood they're going to get some disease.
I was going to do that.
Yeah?
Yeah.
So you can, I mean, there are two ways of doing it. You can do the simple, the 23andMe, where you get a few tests. But I think what will happen over time is two things. More and more people will get the whole genome sequenced, because many of the variants you won't pick up in the simpler 23andMe test. And that cost of getting your entire genome sequenced is coming down and down. Mm-hmm. The second thing that needs to happen is for scientists and doctors to understand what are the risks associated with some of this variation, because most of the time we don't know that yet.
Okay.
And that's coming, but that obviously takes research to understand how these very tiny changes in DNA can affect levels of production of a protein or the ability of that protein to do anything. So that's a lot of research is trying to understand this variation in the human population.
What about in terms of healthcare and insurance? You know, if your insurer knows your DNA sequence, and research comes out that if a person has this type of, they're more likely to get this type of disease, where does it fall into?
Well, this is a big question, right? And this is why there are headlines when big tech companies like Google suddenly have access to lots of healthcare data. That was a story about that fairly recently. Mm-hmm. There is in the US an act called GINA, the Genetic Non-Discrimination Act, which says if you are a health insurer or a life insurer, you are not allowed, or an employer, you're not allowed to discriminate against someone based on their genetic sequence. So, because absolutely you can imagine the scenario where you have some gene variant that makes you very likely, and again, all of these things are probabilities, which makes it complicated, but you're very likely to suffer from some terminal disease. Why would I, as an insurer, I would raise your premium? But that would be, currently, that is genetic discrimination. So right now, that's not allowed to happen. The question, of course, is who will know what in the future? Yeah.
How good are we actually? How much do we know about our DNA and what we can do with our DNA? Because I hear a lot about CRISPR and what they're doing, and the mountain of research that's coming out of this. Are we really close to understanding every single gene and what it does in our body and how we can use that?
No. We're moving in that direction, right? Ever so, the Human Genome Project, that was a major undertaking to get the sequence of the entire DNA, but that was one or two people's DNA. Yeah. And so then you start looking in individuals and trying to understand the variation and what changes there are. I think the biggest challenges are, again, a couple of big challenges. One is there are many genes that we don't know what they do yet. And there are many changes, not even necessarily within the genes, but nearby the genes that decide that or control how much of that gene you're actually going to produce. Mm-hmm. Those are very hard to figure out. And then you put that into a grid with 20,000 other genes, and you have to start to look at the combinations. Because there are lots of what we call single gene diseases, where having a single gene mutation will predispose you to a particular disease. Things like breast cancer, that's very well established. The more complicated traits are controlled by many genes interacting. And so that is much harder to pull out and understand which of those genes, how they interact, and whether they're kind of additive or synergistic or even cancelling the effect of the other variant. And that this starts to become an enormous kind of data challenge.
Yeah. And do you ever think we'll reach the point where we can pinpoint the gene and eventually cure it from a gene level rather than more, chemotherapy or other various curing types?
Yeah, so I think, you mentioned CRISPR, this way to edit DNA. Mm-hmm. And And that's coming. I mean, there were some babies produced.
I've heard, yeah.
Right?
In China?
In China.
Yeah.
And, I mean, the scientist involved has gone to jail for a few years. So this was not...
ethical.
This was not sanctioned.
Okay.
And it was done all under secret. He wanted to make a big splash. As with many of these stories, there's often ego involved. So we're moving in that direction. And again, with CRISPR, the question is, what will we be editing? So if you edit, if someone, I mean, I've even heard the idea that in late stage cancer, you might edit out some pain receptors. So people will still be sick, but maybe without the pain, and maybe that would be a better way than taking some of these terrible drugs that really kind of just block everyone.
Yeah.
People are not themselves on these drugs. That's one level, what somatic gene editing. Yeah. The bigger question and the controversy with this scientist in China was he was editing the germline. So he has taken out a piece of one gene which functions in the immune system, and every cell in these children's body has lost that piece of the gene, and so that will be passed on for generation and generation and generation. And so that is totally changing, not totally, but that is modifying the human genome permanently, as opposed to just treating someone somatically.
And what does that germline do?
So that germline, so if these children, when they have children, they will pass on that altered, that CRISPRed gene.
But what is this function? What is the functionality of this, him taking it out? What?
So the idea was that this was a very, if he took out this piece, these children would not be susceptible to HIV.
Okay.
Which is a noble idea on the face of it. Yeah. And my understanding is that these were going to be the children of people suffering from HIV.
Okay.
So on the face of it, it looks very nice. The trouble is, he didn't create the same mutation that is known to be protective from HIV. He almost invented his own change. And so no one knows how protected this will be.
Okay.
And then that will be passed, because that's in every cell of these children, will be passed on to their children, and you keep going down the generations.
So we're nearly there.
So it's been done, right?
Done.
The human race has been modified using this CRISPR system from bacteria. Right now, it's done in very few people.
But what are the implications? I know it's been done now, then the babies. What about in 10, 15, 20, 30, 40 years, when the real signs of its effectiveness is probably going to be shown?
I don't know if you've seen the movie Gattaca.
No.
It's a science fiction movie set. It was actually made, I think, in 1997. Yeah. And it's set at some point in the future, but it actually has two classes of people. The genetically modified, and I'm blanking on the name of what they call the kind of natural birth, what we consider natural birth. And you've actually, society has moved in this direction where people really they select the genes, essentially, or the traits that go into their children. Yep. And so these people are beautiful and tall and highly functional. And then there's this underclass. So it's a beautiful movie as well as scary, but it also it's a very interesting ending. I'm not going to spoil it for you. But that's potentially where we could go. So whatever inequality we already see in humanity could get exaggerated, because these therapies initially will not be cheap.
Yeah. Eventually, it will be cheap.
Eventually, it will be much cheaper. But it's still a question. It will always be a question whether we know enough to really do this. Yeah. Because there are all kinds of amazing people who've had single gene mutations and lived fantastic lives. We're from Albert Einstein to Abraham Lincoln. There's many, many more that I'm now forgetting, but many of these people had different syndromes that, on the face of it, if we were genetically modifying, we might have removed that gene variant, or even not even selected that embryo to grow, and then we would have lost these amazing people.
Yeah. How does epigenetics fall into this? does this make it more complicated in studying the effects of gene and the expressions?
So epigenetics are the modifications on top of the DNA.
Yeah.
So they are changeable, reversible, but they do affect the amount of a gene you produce. And so if you look at identical twins, for example, as they grow up, they're exposed to slightly different environments, and so they don't look as identical as they used to over time. And part of that is because the environment they're in changes their epigenetics, the food they eat, changes the epigenetic code on the DNA, and so things change a bit. So that it's another layer of complication.
How much of variation does that put into place? Or is it, because I'm trying to understand, it's like, okay, you have your genes and then you have these epigenetics which determine how much you're producing of these genes, but to what degree is it fully controlled by these epigenetic things, or how can we see them?
You want me to give you a number, right, on a scale of 1 to 10?
More of like to just get things into context, because it's kind of like very complex right now.
It is, absolutely. And that the epigenetic modifications on a gene will actually be different in different tissues. And some of that is deliberate. Some of that is what makes your liver cell makes liver enzymes, and your muscle cell make muscle proteins, and not the opposite. But then there are tweaks on that where the muscle proteins will be, the genes encoding those proteins will be on at different levels in different people. And some of that is due to epigenetic. I'm going to be very vague and use the word some, and that will change as well. And, all this research in biology, I've heard a lot of stories about longevity, and there's more and more research into, creating, some form of, drug or some form of genetic that can enable us to live a lot longer than we normally would. What's your opinion on this? Well, we're doing really well already.
Yeah.
If you look at life expectancy now versus 100 years ago, it's very approximately doubled. And so, yes, there's a lot of interest in two things. One, extending lifespan.
Yeah.
The other one, which I think is probably more exciting, is kind of extending wellness, so that you could actually live maybe a few more years, but you would actually be really healthy in those years. And there are certain populations in the world where there's very high incidence of people that live over 100 years. In and they tend to be in small isolated communities, and the question of course is, do they have some special longevity genes, or is it their environment, or is it what they do, how they live? And of course it's a combination of the two. The estimates are it's really not so much in the genes, it's more about the diet, how they are kind of emotionally and mentally, and kind of often lack of pollution in some of these areas.
Yeah, I've heard of this island in Japan.
Okinawa.
It has nearly everyone's over 100 years old, which is very strange. But Japanese people do eat very, very healthy, I assume healthy meals, and they often, sleep well and socially really well. So all these factors play a role in how we express certain genes. So it's
hold on. No. I don't think we know enough to make that statement, because there's other things, and that Okinawa community is interesting. Yes, there's the diet.
Okay.
There's also how much they eat.
Right.
So they tend to undereat. They have a phrase for stopping eating when the stomach is 80% full. Not when it's full, but little. And they let that feeling of satiety, they just wait, and that's fine, they're done. And they tend to eat on smaller plates. There's all these kind of little tricks that get them to, I guess, restrict their calories, restrict their intake a little bit. But then there's other things which are much harder to quantify, things like sense of purpose. Like, people don't retire. They just keep going and they do different kind of things, but there are people on that island that still fish for their family, even at the age of 100.
Gee.
And then they have these communities where I don't know if it's specific to the women, but certainly the women are in these communities where they've kind of been looking after each other all their lifetimes. So they have this very close-knit community. And if something happens to one of the ladies' families, the other one will help out. So they have these tight social bonds. So that's why I kind of stopped you about saying how this affects gene expression. It probably does, but I wouldn't say we know anything about it. And we're not going to go into these poor ladies and look at what's happening in their brains, which is where we would need to look at some of this. Mm-hmm. But there's no doubt that some of these factors, at a very simplistic level, that sense of community will protect people from depression, will keep them active, will keep them engaged in the world. They have this idea of a sense of purpose as opposed to retirement. So longevity, I think, is much more complicated than a pill. Yeah. If you were miserable, a pill can help to a certain extent. Maybe it can get you out of the hole, but it's not. That kind of a lifetime of misery will have its effect on the body.
Because I was listening to a podcast and they were talking about this, like depression and anxiety, and they were saying that the pill, the drug doesn't really do much. If you have to change the way you think about things and the way you frame things. These are what really helps, and improving your life on a very practical level rather than just taking a drug does more than getting chemicals in your body. How much do we really know about depression? These drugs that treat depression and anxiety, and are they really effective, or are they some form of what are they called?
Placebo.
Placebo, yeah.
It's a great question, and I'm definitely out of my comfort zone here. But I remember hearing an analogy of, and this is from a scientist studying these mood systems, and he studies it in mice and in fruit flies. And his analogy for some of the drugs that we use is it's a bit like kind of opening the hood of the car to see the engine and just sprinkling oil on at random. Because when we take these antidepressants, they affect all the neurons that are using a particular neurotransmitter. Yeah. So right now it's a very crude tool. Having said that, it seems to work for some people. But it is also trial and error. You try one drug and you have to try it for a while, a month or so. If you see an effect, great. But many psychiatrists will tell you that it's only part of the solution. Right. And that with depression or anxiety, that exercise is fantastic. Exercise can be just as good as the antidepressant. Therapy in combination with all of this to understand what you're going through. Eating well, sleeping well. We come back to circadian stuff. So there's a whole package. And then if you do all of that, you're never going to be able to say which one was the key thing. Yeah, that's in a way it doesn't matter as long as people can be relieved of this. People talk about meditation. I mean, people will try anything, and sometimes maybe the drug allows you to get up just a bit, and then you're kind of motivated to try a little bit of exercise, and it all works together. Yeah. I think we always want the magic one thing in the same. I love this idea of the pill that could make me live forever, or the pill. People also talk about the pill that would give me all the benefits of exercise without actually bothering to exercise. But what's the point?
Exactly. It's very interesting. You're talking about the complexity of these things we're talking about. They're not very black and white. They're not clear cut. There's a lot of nuances and these processes. But nowadays, like when you're, on Facebook or, you're listening to someone, they make it very binary as one thing works for everyone. And how can people sort of see pseudoscience and understand when something is very, on the edge of pseudoscience, and which one is, very science and clear cut and researched?
That's a great question, right? Because most people don't have either the ability or the interest to go and read the primary literature and understand the complexities. And you can even have two scientific papers sometimes coming to opposite conclusions. So there is debates within the field. I think anytime you see a really simple answer to anything, you should be a little suspicious. Yeah. Often these things you might want to try yourself. And again, it comes back to the individual differences. Some things may be fantastic. You might love eating for 12 hours a day. Your friends might love just being 8 hours a day, and you may say, well, it's not worth me going down beyond the 12 hours. So in a way, and then you make decisions, right? There are things that we do that are not necessarily good for our health, but we do them anyway because there are other benefits.
Yeah.
You know, having a dog, for example, is not an economically viable, smart idea at all, but there are lots of positive benefits that come from having a dog. Yeah. Gets you outside, gets you walking, companionship, all of that.
Yeah.
So not all of our decisions are rational, but some of them are positive.
Yeah. Like, why I asked this question is, like, when, living in, like, Afghan household, and you go to, dinner parties, and then you sit around and everyone's, like, giving people advice about, which medicine they should take and what it's done for them and how it worked for them, or, certain diet tips. And then you ask yourself, you say, okay, like, they're not doctors. They probably haven't looked at the literature, and the advice they're probably giving is probably, for them it's worked, but it would have worked for others, and I think a lot of people are not very sort of cautious when it comes to this. And in, the age of fake news, and when you see article after article saying, a scientific study proved this, and you automatically assume that it's true without really looking into the, scientific literature and seeing, okay, what is true? How can we understand it's true? What has been done? What hasn't been done? So it's very interesting. What about your own research? Where are you going in for? What are you doing now, or what are you looking at?
So we are using these neurons in the fly brain that have these clocks, the circadian clocks. We're using them almost as model neurons to understand some general properties of how neurons work and how the brain works. So one of my colleagues had discovered a while ago that these neurons actually change their structure. This is neuroplasticity, which is a buzzword all over. Everyone wants to keep their brain plastic into old age and stuff. But these neurons actually change their physical structure with a 24-hour rhythm. So if we look at the tips of their neurons, they're expanded in the morning, and then they've retracted in the evening, and by the next morning they've expanded again. And so this is a beautiful system to study the basic phenomenon of plasticity of neurons.
Okay.
And so these neurons, so the questions we're really interested in are, how are those changes being driven? And what happens with those changes? Are they then making and breaking connections to downstream neurons? And can we use that to understand how that happens in the first place? Because these are such fundamental properties of neurons that are very hard to study in other systems.
So it's mainly focused on neuroplasticity. And from what I understand is that it shifts between how plastic it is is it a rhythm, or?
It's a rhythm, and so it's a rhythm of the, imagine my hand, right?
Yeah.
So this is the neuron in its expanded state, and this is the neuron going down to where the nucleus is. So these are the tips of the neurons, and they're expanded in the early morning, and then they retract in the early evening, and then they expand again the next morning. Okay. And so what's from the biology of the system, this is amazingly predictable because we know when these changes are going to happen. If you think, neuroplasticity is meant to underlie memory, for example, right? But if you imagine trying to go into a complex brain and find the few neurons that have made new connections that encode for a particular memory, that's almost impossible to do. And so you can't really understand the basic properties of how those neurons are finding each other, how they know which connections to make. So we can use this as a simple system to understand the basic biology.
Right. And would this ever, relate to human beings and how our neuroplasticity neurons work?
This is the thing, we never know. We think so, but we never know. Yeah. But the idea is, it's very similar kind of from the way back the early days of circadian rhythms. We can study this in a simple system like the fly brain. In fact, even simpler. So our logic is, let's study this in clock neurons of the fly brain, and then let's look in other sets of neurons that are known to be plastic and see if these similar molecules that regulate the clock system plasticity are regulating other parts of the fly brain. And if that's true, then it makes sense to then look in mammalian neurons in some way.
Yep.
We will never go in and look at a human brain in that way. That's certainly not my lab.
Yep.
But we might have kind of no genes in the pathway that could be important to look for, say, in autism or schizophrenia, where there are definitely defects in neuroplasticity. These could become, we could help identify perhaps risk factors for those diseases. That's now I'm really extrapolating.
Yeah.
But at a fundamental level, neurons send signals, they fire, they communicate to downstream neurons, whether you're in a fruit fly brain or a human brain.
Yeah.
So some of these properties should be general.
So it's interesting, neuroplasticity as a subject to study, because when you're learning about how to learn and what's the best effective types to times to learn or what to learn and how to learn it, this word neuroplasticity often comes into play. And why I say this is because a lot of people think that as you get older, you can't learn as much. And is this to do with the amount of neuroplastic neurons you have, or what's the correct way to frame this or look at it?
It's a good question. I'm thinking.
Yeah.
I have more to learn about neuroplasticity.
Yeah.
But I would say I'm going to have to pass on that question.
Okay.
But I think there are definitely kind of phases in human beings when the brain is undergoing tremendous plasticity, when we're learning language. But now we're going back to kind of very young ages, 2 and 3.
Mm-hmm.
There are also tremendous changes that happen in the brain in teenagers and go on into our mid-20s. After that, there's less overall changes, but we still have the capacity to learn new things. We just sometimes, yes, it may be harder, it may take longer, or you may have less motivation. Things are generally more hardwired as we get older, but people can still learn new languages. People can still do things if they have the right motivation. In terms of actually measuring plasticity, again, this is where human beings are not such a good test subject, because you can't go and measure something in a 3-year-old and take a piece of his or her brain. That's not the way we do things.
Yeah, of course. Thank you very much, Justin. Really appreciate your time for being here.
You're very welcome.
Thank you.
Thank you.